Hi, everyone. I'm student Dr. Hiremagalore, and my mentor is Dr. Ehab Sarsor, and our project is HEF1 alpha dependent lipolytic signaling regulates metabolic plasticity of aging normal human fibroblasts. So the subject of my project concerns a growing clinical problem. And as you can see from this graph, the US elderly population has been rapidly increasing over the recent years and is continuing to increase. And this is a problem because until the age of 45, the leading cause of death is unintentional injury. However, after the age of 45, chronic diseases become the leading cause of death, which shows that something is changing with aging. And to investigate this problem, our lab studies cellular aging. The two types being replicative lifespan, which is when cells divide until their telomeres shorten, and chronological lifespan, where normal cells stay in quiescence and divide when they need to, which is what this project focuses on. Recent data from our lab showed a link between altered lipid metabolism and aging in normal human fibroblasts. This was done through RNA sequencing to measure global gene changes. And the top portion of image A shows young cells, and the bottom portion shows older cells. And you can see that the red, which is downregulated genes, and green, which is upregulated genes, don't line up between the young and old NHF cells, showing a contrast in gene expression. Bioinformatics analysis, which is shown in B, found lipid metabolism to be the most affected pathway with the highest number of gene changes when comparing old versus young NHF cells. And one of these genes that changes with age is HEF1 alpha, or hypoxia-inducible factor 1, which is a transcription factor known for its role in angiogenesis, but it has also been found to play a role in cellular metabolism, and in our lab, lipid metabolism as well. This figure from our lab shows data from a real-time PCR assay, where you can see a significant decrease in mRNA levels of HEF1 alpha with NHFs as they age, as well as a significant decrease in mRNA levels of HEF1 alpha in NHFs from the same individual as they age in B. But HEF1 alpha is actually most regulated at the protein level, and this data led us to the hypothesis for our project, which is HEF1 alpha regulates lipid metabolic plasticity of aging normal human fibroblasts. So for our experimental design, we had quiescent primary cultures grown in 4% oxygen, which is the physiological level of oxygen. And we had 20 different cell lines ranging from 17 years old to 79. And we also accounted for sex and race as well. Our goals for the project were to measure HEF1 alpha protein levels in all the different cell lines, measure lipid droplets in the cell lines as a biomarker for changing lipid metabolism, and to measure the changes in cell proliferative potential. Our results from two independent Western blots showed an age-associated decrease in HEF1 alpha expression. From left to right, group 1 shows young to old NHF cells, and group 2 shows old to older NHF cell lines. Both groups show a very clear trend of decreasing HEF1 alpha protein expression as the NHF cell lines increase in age. And to see how this decrease correlated with lipids, we used live cell imaging. We used Hoysch to stain the nuclei blue and Bodepie to stain the lipid droplets green. And these three groups are representative of young, old, and older NHF cell lines. And you can see a clear increase in green fluorescence with age, which represents lipid droplets. And this is the quantified data from the previous images from live cell imaging. And you can see that there is a similar age-associated increase in lipid droplets. Lastly, we also examined cell proliferative potential using quiescent cell lines by subculturing them in low density and measuring the growth index. You can see a clear difference between the red group and the blue group. The red group is cell lines over 50 years of age, and the blue group is cell lines under 50 years of age. And there's an age-associated decrease in cell proliferative potential based on this data. So in conclusion, our data shows a decrease in HEF1 alpha protein levels during aging in NHF cell lines, And these changes in HEF1 alpha were correlated with a decrease in cell proliferative potential and an increase in lipid droplets in aging NHFs as well. And for the future, we would like to test the causality by manipulating HEF1 and then measuring its effects on cell proliferation and lipid metabolism. And we would also like to measure protein levels of downstream targets of HEF1 alpha, such as G0, G1 switch gene and ATGL. And yeah, and I would like to thank everyone from Dr. Sarsour and Dr. Zady's lab, as well as KCU, for allowing me to do this research. Thank you.
Questions, anyone? I have to say, I just want to make this comment about all of you. Since the summer fellowship at the end of the summer, you all have cut down a lot on the verbiage and increased your images and pictures, and you're getting through much more faster. It's really interesting. So you become more concise in your presentations, which is a good thing. Okay.
I know we're trying to stay on time, so just a really quick question. For those of us who are past that early stage on your time span, I'm wondering, are you aware of anything that we might do to increase HIF1 alpha for ourselves?
Sorry, can you repeat the question?
So I won't repeat it all, but I'm wondering if you're aware of anything dietary, exercise-wise, things like that.
Increase HEF1 alpha.
Yeah, just to get it back up there where it should be.
I don't know of any dietary or exercise changes you can make for that, but I feel like that could be something. I know there are HEF1 alpha like protein inhibitors, and there are things that can enhance it that you can take, and that was going to be the future of our research, to test the causality with those agents.
Dr. Biddle, expose yourself to hypoxia. There's a chamber where you can reduce the oxygen.
Or you can move to the top of a mountain.
There you go. Michael Jackson had, an oxygen cylinder.
Okay, guys, keep moving. You guys are funny. Okay.
Well, the other thing is, if you come up with that idea, that's a business in waiting, right? A multi-billion-dollar business in waiting. So,
remember, guys, the downregulation also in the RNA level. So there is something upstream of F1.
Yeah, sure.
Transcription is there. So I think it's called NRF1, NRF2, which is another transcription factor that may affect HEF1. So we're still in working, and I know Spoorthi is excited about the drugs that we might be using. So yeah.
Okay, thank you very much.
For you entrepreneurs out there, there's an idea worth pursuing.
Yes.
And as Dr. Biddle was trying to say nicely, yeah, there are folks of us who might invest in that or pursue that or purchase that. So,
well, Dr. Biddle, I can tell you for definitely with scientific references, one thing is olives. Olives have a secret. Okay, they are very rich in an antioxidant called hydroxytyrosol. And if you look at my publication, you will see a lot of things I'm talking about it. So you'll like it. It's delicious.
Dr. Sarsour is starting the olive business, certainly.
All right, you guys, it's time to move on. All right.
I know someone who's been helping import the olives.
Yeah.
Okay.